I'm in San Francisco now. I'm at a blood bank, actually, that's affiliated with UCSF. So I spend about half my time at UCSF and half my time with people doing transfusion medicine research. And who I am, I was Doug's PhD student way back in the day. As evidence there, I'm sort of hiding in the back there with a cup of something. I'm not sure what. Certainly look like riffraff, huh? And magically, Doug looks about the same. It's been like 2 decades. He looks pretty much the same, which is kind of sickening on some level. And I should also say how I met Doug initially, right? So this is definitely, I think we all have these moments in life that are sort of like human pinball moments where you feel like you have very little control over what's happening and it totally defines your trajectory for good. One of those moments was meeting Betty Korber, who was my mentor before Doug. But what happened, I was here, I was a rotation student, and I was actually about to drop out of grad school. I don't know if you remember that, Doug, but I was actually dropping out, which would have probably been better for the world, actually. But I was dropping out of school. I felt kind of disillusioned by my rotation experiences, and I talked to Betty Korber, and she said, well, you know, you're not working on HIV there in your rotations, and you loved working on HIV so much, maybe that's part of the problem. Why don't you go talk to my friend Doug Richman, you know, he's over there. And I was like, all right, and I'll go over there. And so I remember very clearly walking into Doug's office and explaining my situation to him. And I remember very clearly asking him this question. I said, well, you're not listed as a faculty member in my graduate program, so could I actually do my dissertation here when I'm done? And he kind of looked at me. These are the words that came out of his mouth. The words that came out of his mouth were something like, if there's mutual interest. But what he really meant, this is like, for me, like the subtext was, I'm goddamn Doug Richman, I can do whatever I want. That's what it came across, like, this guy's cool. And then he tolerated me and the rest is history. All right. So I just wanted to share with you guys a couple of quick slides about Doug-Derived Wisdom that he offered me back in the day that I share with my own mentees, now that I'm driving my own Motley Crue school bus. But Doug always used to love saying this, and it's actually very meaningful to me. So, I'd walk in all excited, be like, Doug, subtype B has a glycosylation, a glycan at like position 308, but in subtype D, it's at position 309. And, he'd be like, what does that have to do with the price of eggs? Like, telling me something I should care about. And I love actually saying that to my own mentees. The other thing, I don't know why I saved this one. This one really sucks, to be honest. But Doug, and I'd be like, hey, Doug, you're going to be at Journal Club? Or something. He'd be like, you bet your bippy I'm going to be there. And I literally say that to people in my lab. They're going to be like, hey, they'll say something like, are you going to be at my thesis committee meeting? And I'll be like, you bet your bippy I'll be there. And they look at me like I'm like 250 years old. This one hasn't come in handy as yet, but I'm pretty sure it will. I'm pretty sure it will. Who here in the room remembers this Doug quote? Anybody? Davey, do you not remember this? Matt, you remember? This was actually, Davey, this was your work in progress. And you were talking about people like, like a recruitment issue in this like study. And Doug was like, just very, like matter of fact, you can't back out of a prostate massage. I really love it. And last but not least, for those of you who have actually followed my career, this is the last thing he said to me when I left his lab. He said, do not ever publish anything after you leave my lab. If you must, make sure it happens infrequently and insignificantly. And Doug, I've kept my word, man. I've really kept my word. Okay, so I'm going to start off with a very rudimentary, maybe even an insultingly rudimentary figure that I've designed to just explain why antiretroviral therapy doesn't result in a cure, which I think is background knowledge to everybody here, but it's a good way to start out. So if we think about untreated HIV infection, we have a population of HIV-infected cells in the body. They pump out virus. Those viruses encounter uninfected cells, and they generate new populations of infected cells. And in the absence of any small molecule drug treatment, this process continues ad nauseum. But in the setting of antiretroviral therapy, essentially you just delete the whole right side of that schematic. So there's little or no ongoing replication during treatment. But what we do know is there's a large or some sized population of HIV-infected cells that pre-existed the onset of therapy, and none of the drugs that we take at this point do anything to destroy that population of latently infected cells. So these long-lived latently infected cells tend to be invisible to the immune system and may survive for years during ART, so when you stop treatment, the virus comes back. So key questions about the latent HIV reservoir, and this is kind of what about half of my lab focuses on. So one thing is, how do HIV latently infected cells differ from other cells? What are the natural host determinants of HIV reservoir size, which is very closely aligned with the stuff that Huldrych just presented, although we use different inputs. And another thing, we do a bit of diagnostic development as well. How can we actually estimate the size and the anatomical distribution of the latent HIV reservoir? But what I'm going to talk to you about today is something that we've really started shifting our energies to in the last couple of years, and about half my lab is working on this. Besides the observational stuff to understand the reservoir and look for predictors of reservoir size, we're really trying to develop a range of techniques to selectively destroy latently infected cells to achieve a cure. So there's several different approaches that are being discussed at this point. There's treatment intensification, and as Huldrych mentioned, that's kind of the only tool that we have that's road tested to any extent. When we start treatment early, we can see that there's smaller reservoir size. Then there's this group of approaches called the shock and kill approach, where we try to promote viral expression to make infected cells immunologically visible. There's a range of immune-based therapies, therapeutic vaccination, and then last but not least, there's gene therapy. And gene therapy efforts against HIV in the last few years have really taken off. And I think there's really one central reason why they have. There have been major advances in gene therapy which give us a new hope for a cure. So if we go back to, for instance, when I was a graduate student, if we wanted to design an enzyme to specifically target HIV or target a receptor that HIV used, that was a really, really tall order. But really, after the discovery of the CRISPR-Cas9 gene editing system, which is essentially an adaptive immune system in bacteria, it really makes that a lot easier. It's a lot easier to design an enzyme that can specifically find one genetic sequence. And so most of what I'm going to present to you today are from this handsome bunch over here. So on the left, we have Leonard Chavez and Kyle, who we call Baby Kale Raymond, who are mainly working on developing viral vectors to target HIV. And on the right, we have, excuse me, we have Mohammed Bouzidi, who is using what we call CRISPR-Cas9 ribonucleoprotein complexes to target HIV infection in primary myeloid cells. But I'm just going to focus on the work of the folks on the left. So I just casually mentioned that the CRISPR-Cas9 gene editing is very, quote, easy. So this is how it works in a nutshell. If you happen to know the sequence of your gene of interest that you're trying to target, whether it be HIV or a HIV host dependency factor, you design this thing called a guide RNA that's homologous to the target that you're trying to destroy. And then you combine it with this Cas9 nuclease. It targets it, it cuts it, so you have a double-stranded nick, and then the non-homologous end-joining DNA repair mechanism within the cell stitches the DNA back together. And not every time, but a good chunk of the time that happens, you get a mistake. So you get an insertion or a deletion, and you no longer have a viable sequence. And so this really provides a very convenient platform to target and silence specific genes. Another thing that I think is important, so in my head at least, I think there's two basic classes of gene editing approaches that are being discussed. So on the left, we have ex vivo gene editing, and the idea here is that you can remove cells from a patient, you can genetically tweak them however you want, and then you can infuse them back into the patient. And on the right, this is something which I think people find are more violating, upsetting, and shocking, although I think there's been a lot of movement on this front in the last couple of years. But you can also have in vivo gene editing, where you send the gene editing vehicle directly into an individual to alter a genetic state. And in terms of how this would be applied to HIV, on the left, we can make cells resistant to HIV, but for instance, by getting rid of the co-receptors that HIV uses to enter cells. The other thing we could do, for instance, I'm sure everybody here has heard of CAR T cells. We can create super potent anti-HIV immune cells. And on the right-hand side, in terms of in vivo editing, I think the most obvious application of this for HIV is direct targeting or excision of the HIV provirus within infected cells. So we're certainly not the first lab to do this. There's been a range of efforts that have already been published, and I'd say the most seminal work has come out of Kumail Khalili's lab at Temple University and UPenn. And so they've done a lot of proof of concept kind of work, but there's still a lot of challenges left to deal with before we can really test this in a clinical context. So this is by no means a comprehensive list, but I'd say these are the main obstacles facing using CRISPR-Cas9 as an eradication strategy. So the first thing, and I think we heard a lot about this on the TV and radio with the CCR5 babies, but we do know that these enzymes, as amazing as they are, have an off-target rate. So some fraction of the time, they're going to create some sort of genetic mistake in a gene or location that you don't want that mistake in. And if that mistake winds up in the wrong place, that could obviously be really deadly. Another problem, and this is a slide that I've sort of corrupted from Betty Korber. I'm sure many of you have seen some version of this slide over the years. But on the left, that's a phylogenetic tree representing the global diversity of HIV. And on the right, that tiny little blob represents the global circulating diversity of influenza. And we know even that is a challenge for vaccine design. So we have orders of magnitude difference here. But we have to design guide RNAs and a gene editing approach that can tolerate that much genetic diversity. And then lastly, related to the last point, is you can have escape mutants. And there are already a couple of papers that have shown with these HIV excision strategies, if you hit the virus at one place, the virus, it selects for mutations that are resistant to your guide RNA and you're back to square one. So here are the things that we're doing to fight this problem. We're actually doing a range of things on the off-target editing front, but one thing I'll talk about today is using alternative enzymes. So there have been alternative versions of the Cas9 enzymes that have been created that have much lower or really non-existent off-target editing activities. And it's kind of similar to like high-fidelity polymerases as opposed to like standard Taq polymerase, which is a little bit sloppy. On the viral diversity front, we're using a bioinformatic approach to identify very highly conserved guide RNA target sites within the HIV genome. And on the escape mutant front, we're taking an approach that kind of mirrors really what's happened with small molecule antiviral therapy over the years. If you hit the virus with one drug, you're likely to quickly breed resistance. You hit it with two drugs, probably still going to have an issue with resistance. But if you hit the virus with 3 or more drugs, it seems like the virus can't slither past that hurdle. And so we're using a similar approach to simultaneously target the viral genome at multiple locations. So here's the basic workflow. Again, it all goes back to awesome mentorship, but I relied on my former mentor, Betty Korber at Los Alamos National Lab, to design guide RNAs that target very conserved sites of the HIV genome. And so what we did essentially was we adapted her vaccine immunogen algorithm. So Betty is very famous for designing vaccine antigens that have maximum coverage across the HIV proteome. So we took that algorithm and we just tweaked it a little bit to find guide RNA targets instead of proteomic targets for vaccine design. By surveying a massive population of sequences, over 3,000 sequences in the Los Alamos database, we were able to find hundreds of guide RNAs that fit our criteria. So once we found those things, and I skipped one step, we actually test the editing in a test tube before we even get to cells, but almost everything works in a test tube. So the next thing we do is we test each one of these candidate guide RNAs in JLAT cells. And so these JLAT cells are immortalized CD4 T cells that contain a version of HIV that has a GFP reporter. And when the virus becomes reactivated within the cell turns green. And so what we did was we stably transduced these JLAT cells with a Cas9 gene, so they're just constantly cranking out the CRISPR-Cas9 protein, and then we just in vitro transcribe guide RNAs and just nucleoffect them in. And then we use this procedure called tracking of indels by decomposition, or tide analysis. And Joe, do you remember Rodin Parada? Remember him? But we tried to develop a version of this years ago, but our purpose was to look at length polymorphism in the HIV envelope. It's the same exact principle. So you can essentially detect phase shifts in the chromatogram, so you can use bulk sequencing data to identify insertions and deletions. So this is the most common way that CRISPR-induced mutations are measured. So what we see is that out of the 30 top candidates that we tested, 6 of those very potently induce indels within the HIV provirus, and those are represented by the tick marks that you see there on the HIV genome map. And what we were very pleased to see is that all of these sites either are absolutely conserved across the HIV genome, the whole M group, or at most have one substitution across the entire M group. Okay, so that was all done, again, through this in vitro transcription and nucleoffection thing. So the next step was to design a therapeutic lentiviral vector. And so what we did there, we designed actually a whole family of vectors that have restriction sites so that you can very easily swap out these guide RNAs and different enzymes. But we started out using this rationally engineered Cas9 nuclease with a very low off-target rate. Once we designed those vectors, we produced lentiviral particles, and then we tested these individual therapeutic lentiviral vectors in a new cell line that we developed called JLAT enter cells. So why did we do that? So I mentioned that these JLAT cells, which were designed in Eric Verdon's lab at the Gladstone, they all harbor a latent provirus. Each cell harbors one latent provirus with a GFP reporter sequence. The one problem with them is they don't express envelope, so there's no infectious virus produced by these cells. So basically, we can't use this cell line to look at the effects of our gene editing on actual infectivity of the virus that's produced. So we stably transduced these cells with envelope. We restored envelope expression to the cells so that they now produce infectious virus. So we can directly assess the effects of our gene editing on the viability of the viral progeny generated by these latently infected cells. And now, using this system, we have 4 different signals that we can look at. We can look at directly at the genetic edits. We can look at viral reactivation. We can look at virion production, and we can also look at effects on the infectivity of the virus that's produced. And so what you can see here, I'll just show you one example, that these enhanced Cas9 therapeutic lentiviral vectors inhibit virion production and infectivity. So this first bar graph shows the indels, so the frequency of CRISPR-induced mutations in the cells that were treated. This shows effects on reactivation, and what you can see is the effects on reactivation are pretty modest, which is what we expected, because none of these constructs are targeting the HIV LTR or the GFP sequence, so that should really be retained, and that's basically what we see. What you can see is some of these things very heavily affect virion production. So, a couple of these actually almost completely abrogate virion production. And what we can also see is if we correct for the amount of virus produced, that a lot of these guide RNAs are inducing very significant impacts on the infectivity of the virus that's produced. So all in all, within this model, this shows that this approach works. Oops. Then, in terms of putting this all together, again, to avoid this issue of selecting for resistant mutants, I won't go into detail on this stuff, which is just extremely nerdy stuff that Leonard really enjoys, but he spent probably a year of his life experimenting with all of these different highfalutin multiplexing strategies to pack several different guide RNAs into a single vector. Some work and some don't. I'll show you an example of some of our results. So here we've been able to multiplex 4 of our guide RNAs using something called polycistronic tRNA multiplexing. And what you can see in a nutshell, and this is what we've seen repeatedly, it works. So using this combined vector, we can see edits at all the sites, but we see a reduction in efficiency as compared to each of the mono-targeting vectors, which I guess is not that surprising. But we're optimistic that we can tweak some volume knobs to make this work. Another thing I wanted to mention real quick is that I think a huge problem with this gene editing field is the tools that are used to register and measure the gene editing are super primitive. And the ways that they do it, one, like I told you, is just looking at bulk Sanger sequencing data, which really doesn't give us very quantitative information. The other thing that I've seen in the literature is just an excision band. So people will show like a gel, like a gel image where you can see a band of something that pops out when the DNA is cut, but it's not very quantitative. So we've partnered with this company called Mission Bio to do targeted single cell DNA sequencing. And so we now have a panel that sequences across the entire HIV genome with tiled amplicons. And so now we can actually see if you have linked mutations across the genome. And just to revisit this bulk Sanger sequencing derived data I showed you, here's what the data actually look like when we look at the single cell level. So what we can see is out of 6,300 cells in that run, 1,727 cells were cut one time at one of the 4 sites. It drops to 292 cells that were cut 2 times, and it gets pretty abysmal after that. So only 25 cells were cut 3 times, and only 2 cells out of like 6,000 were actually cut at all 4 locations. And this really wouldn't have been evident using the bulk sequencing approach. So the good news is I think we have a powerful diagnostic approach. The bad news is I think we have a lot of work left to do on this. I'll mention this real quickly. I'm going to gloss over it, but I think the field is looking for ways that we can very specifically activate these vectors in infected cells so that we don't get editing where we don't want it to. So one thing that we've done is we've linked the expression of our vector to TAT. So essentially, just like TAT transactivates the expression of HIV, we've relied on this so that we have a version of the vector that only lights up when it co-infects a cell that's producing TAT, so co-infecting an infected cell. And what you can see in these flow cytometry plots, like on the lower right, is we only get activation of Cas9, of the Cas9 vector, when we infect these, and this is in primary CD4 T cells, when we infect the transduced cells with HIV. And this is an example of the editing levels that we see. So when we actually have HIV in the system, we get 50% of the cells showing some signs of edits. One thing I'll just mention very quickly, anybody here who has done any math modeling, and people like Matt and stuff, if you've looked at any of the data, the whole idea that you could use just a pure reservoir targeting approach to really achieve a cure is almost absurd. I mean, you really have to, kill probably 99.99999% of the reservoir, and no matter how good you are, that's a pretty big stretch. And so one thing that we've been interested in is this idea of coupling reservoir targeting with modulating host dependency factors, so targeting host dependency factors. So in this case, we have this multiplex vector that targets HIV at 3 sites, and then we also hit CXCR4, which this is a proof of concept. I'm not saying that it would be a good idea to target CXCR4 in vivo, but what we've been able to show using this approach is that we can very potently shut down replication in a petri dish. We haven't gone into an animal model with this, but we think this idea of simultaneous editing of both the virus and host could be promising. So to conclude, engineered lentiviral vectors target maximally, that target maximally conserved regions within the HIV genome, effectively destroy latent provirus in cell lines and in primary CD4 T cells. We have looked very carefully, and so far we haven't seen any off-target editing observed at any of the bioinformatically predicted sites. Multiplexing several guide RNAs that target multiple HIV genome sites may provide insurance against evolution of viral resistance, but we definitely need to turn up the volume on that. Single-cell analysis of gene editing provides key insights into the therapeutic efficacy of gene therapy platforms. Limiting vector activation to infected cells may limit off-target effects. And lastly, simultaneous targeting of both the virus and the host axis may warrant, I think really warrants further exploration. Here's my ongoing and future work. So really where we are at this point, which is where the whole field is, I mean, gene therapy always goes to die in one place, delivery. And so it's true, right? And so we're really focusing on how we can deliver these things in vivo, mainly focusing on lipid nanoparticles and engineered exosomes. And then we're also developing relevant preclinical models, because as of now, there's monkey models, which are almost impossible to get your hands on, and you can test like 3 animals. And the mouse models all pretty much involve engraftment of human fetal tissue, which is becoming very, very difficult to do. I'm sure you guys, if you read the papers, you can see it's becoming almost impossible to do. So interestingly, my sister is a bioengineer. She was an immunologist, actually she's still an immunologist, but she decided to switch to bioengineering, and she's a postdoctoral fellow at the Langer Lab at MIT. And so we've partnered with her in the last few months to package some of our CRISPR-Cas9 constructs into lipid nanoparticles, which has been super fun. And so we actually flew her out a few weeks ago, and she flew on the plane with all these syringe pumps and stuff. They must have thought she was like a nut job. But she set all this stuff in my lab, and so right now we're actually working with her to use lipid nanoparticles to deliver some of this stuff. Very interesting work that I don't have time to talk about today, but we do a lot of passive observational work with exosomes, which are a specific type of extracellular vesicles that are involved in cell-cell communication. We're kind of going past the biomarker discovery phase with exosomes, and we're working with this company called System Biosciences in Palo Alto to engineer the surface of exosomes so that we can steer them to particular tissues or particular cells expressing, surface ligands. And so what we've been experimenting with now is taking some of her surface engineered exosomes and packaging them with both cytotoxic cargo as well as our CRISPR reagents to target HIV. And last but not least, Hanna Sperber, who's really an amazing PhD student in my lab, has done some really cool work to develop a preclinical model for HIV cure strategies and specifically for gene therapy. And this idea really came about just through drinking too much beer at a journal club. We were like, well, why instead of using stem cells or using fetal tissue derived cells, why don't we just take these JLAT cells that we're playing at in petri dishes all day long and just do tail vein injections and see what happens? And it turns out it's really blossomed into a really nice project. The JLAT cells engraft tremendously into lung, spleen, bone marrow, and peripheral blood. And now we could treat them with TNF alpha and other latency reversal agents and show that the cells go from being non-fluorescent to glowing green. And so now we're coupling that with this single cell sequencing strategy, and we're hoping that this will be the test bed that we need to efficiently test the vectors that we're developing. Okay, I'll close with that. I'd like to thank all the folks in my lab, and specifically the folks in Boldface are the people who did this work, and our range of collaborators at our blood bank, UCSF, Los Alamos, the Marston Lab at UCSF, which is really a center for CRISPR excellence, and some of our industry collaborators. And last but not least, I really had to thank my kids because I had to beg them to allow me to come here and skip Halloween and trick-or-treating, and they very graciously accepted. All right, thank you.